Hello, everyone. In this video, we're going to discuss the different kinds of photosystems that participate in photosynthesis. So this is just a schematic of where we are. We are in the thylakoid, in the chloroplast, and it looks like this. There are stacks and stacks of them, as we described in the previous video. And the easiest way to view this is like the inside out, the reverse topology compared to what we were discussing with the mitochondria. So in this case, light will split water. We're going to get some electrons. They're going to move along an electron transport chain before finding their way onto NADP. And so we're going to generate NADPH, the reducing power. But as the electrons are moving, protons will move in from the outside, which is called the stroma, into the inside of the thylakoid called the lumen. So if you remember in mitochondria, we had high concentration of protons moving into the outside. And then they want to go back inside, and the ATP was generated inside the thylakoid. Instead here, we have protons moving in, so we have high proton concentration, so low pH. The pH of the stroma of the outside is higher. So now these protons, you build this massive, assuming you do a lot of photosynthesis and you have light split in water and so on and so forth. So the electrons are moving and the protons are moving in. Then these protons will really want to go out. Okay, so you stick your machine, your ATP generating machine, and ATP synthase, and the protons will come out. And in this case, also the orientation of course, the protein synthesis machine is reversed from what we had seen in mitochondria. So the ATP generating part, the soluble part, is actually on the outside in the stroma, and you get the ATP right there. So what we're going to discuss today is really the nature of these two bolts of light that I have drawn on the schematic. Let's go back to about 1932. We discussed 1905, how the realization became obvious that chloroplasts are similar with cyanobacteria. So we have this whole symbiosis with it, like we had seen with mitochondria and some primordial bacterium that could do oxidative phosphorylation. But then by the 30s, Emerson and Arnold did a very simple experiment. They just plotted the amount of oxygen you get from every molecule of chlorophyll that you have in plants. And because, of course, chlorophyll, I just mentioned to you, is the key cofactor, the key compound that participates in photosynthesis. That's where photochemistry is taking place. All right. But they found that only one molecule of oxygen is produced from 2,400 chlorophyll molecules on average, right? So most of the chlorophyll molecules are not involved in photochemistry. So this is not where water, this is not where, but in most of them, there is no splitting of water, okay? So instead what happens is they make this antenna, so to speak, which really, that's how it's called actually, the antenna complex. And it's an apt term because it does resemble, so to speak, the satellite antenna, and light will hit one of these green things, which is supposed to be a single chlorophyll molecule. There's not going to be any photochemistry, but the light will move on. The energy of the light will move on towards the reaction center, where the actual photochemistry will take place. Right? So although most chlorophyll is not photochemically active, it does participate in this transfer. And an exciton is something that can transport energy without transporting net electric charge. So this is what these other molecules, the non-photochemically active molecules of chlorophyll, are doing. They are excitons. And the way to view this again is that you have this the antenna pigment molecules. These are all the ones. It's chlorophyll. It's a couple others, and the light hits somewhere, and there is exciton transfer until it reaches the reaction center of the chlorophyll. And at that point, this is where we have a harvesting of the energy. Yeah, and this is where the business happens. There is a little bit of energy loss as fluorescence, but not too much, about 3 to 6%. All right, so what's the reaction that we're dealing with? Is the splitting of water again to get oxygen and 4 electrons and protons. So as we had seen in the reverse reaction, when we're talking about oxidative phosphorylation, it takes 4 electrons for one molecule. Now, Emerson, the same Emerson that we had seen before, I think by that time he was at the University of Illinois at Urbana-Champaign, really in the 50s did some super key experiments and found that there must be 2 distinct photosystems with different properties. So this is what he did. He was flashing light of different wavelengths. And when he did that with wavelengths that were about 600 nanometer, when he gave them a 680 nanometer flash, it absorbed chlorophyll, but no oxygen was produced. So something was weird. And then if he has 680 and 700, it got better. And the 2 flashes somehow cooperate and they didn't have to happen simultaneously. Okay? You could have a lag. So to make a long story short, he came up with the idea that there are 2 distinct, which we know now to be true, there are 2 distinct photosystems. Photosystem II, okay, is the one that we roughly described up to now. This is where oxygen is produced, okay, only by photosystem II. So there is a flash of light, and photosystem II will engage it. We are going to get some oxygen and protons. And what happens with a high energy electron is going to move along an electron transport chain, and it's actually going to find its way on some other chlorophyll molecule. And then a second flash of light, and in the process you get ATP the way we described, with a translocation of protons. Then a second flash of light of that chlorophyll molecule will take place, and then you have again, raising the energy of that electron. It's a high energy electron, and it's going to go through another electron transport chain, and then it will find its way onto an ADP plus and pick up another proton and become an ADPH. So it's 2 different photosystems. So what's the take-home message? There are 2 different photosystems, and one of them splits oxygen, and the other one actually reduces NADP to NDPH. Okay. So this is interesting, and it turns out every photosynthetic organism has photosystem 1, but only, let's say, green plants have a photosystem 2. So clearly, photosystem 2 was an add-on. Okay? And remember, again, it's photosystem 2 that makes oxygen. So you can actually have photosynthesis, just photosynthesis, without releasing oxygen. Okay. So, and we'll see how these organisms here that don't release oxygen, how they actually make their ATP. Okay. Because the way the scheme is shown here in a green plant is that you get some oxygen, the electron gets higher, then you get your ATP through this electron transport chain, and then you have a second flash of lightness in photosystem 1, and then you have another electron transport chain, and then you get your NADPH. So you get your reducing power and your ATP in sort of separate steps. As we will see, you can do with just photosystem 1. This is a great system. It works very efficiently, and we'll see that, but you can do just with photosystem 1 and get both. If not all your electrons, I'm giving you the hint here, go to an ADPH, but since they return through this electron transport chain, so you can actually make some ATP as well. All right, so again, there is no photosystem 2 without photosystem 1. So whoever has photosystem 2 also has photosystem 1. Okay, you absolutely need it to get your reducing power. So it turns out photosystem 2, of course, is an excellent oxidant. It takes electrons from water, and what it does, it transfers them to Q. So again, we have Q reduction. But before we see that, let's see how photosystem 2 looks like. That's the molecule, that's the protein complex that makes all the oxygen on Earth. So it's astounding. Okay, we have an amazing substrate that we all depend on. Okay, we can't live without it, and this is how it's made. All the oxygen that is made, has ever been made on this planet, is made by this complex. I mean, that's quite a statement. So you see a bunch of things. You see all the proteins that's shown here on the classic, schemes. You have the alpha helices, you have beta sheets and loops, and you have a bunch of extra compounds. You have chlorophyll, which is good. That's what you want. But you have all these other compounds, pheophytine. You have a beta carotene. And so we'll see what happens there. And we have a magnesium reaction center that's really, this is where the water oxidation is taking place. So in addition to making oxygen, for a green plant that depends on photosynthesis and photosystem II, if you block it, if you inhibit it, then you got the world's best weedicide. And in fact, atrazine, which is what you see here, is a specific inhibitor of photosystem II. And that's, it's a herbicide, it's a weedicide. And if you see the use, this is, I believe this came from USDA, but if you see the use of atrazine, it shows here the breadbasket, the Midwest, it's glowing. We have a little bit in Texas. You can also see the going down with, along the Mississippi River and so on and so forth. All right, so back to some of these compounds. We have the beta carotene, for example. What do they do? This is how they look like. Of course, we eat a lot of them and orange-looking things like sweet potatoes and carrots. They are antioxidants. I mean, that's a fact. This is how some of the other compounds, how they look like. And you can see the double bonds and all that. I mean, they are really photoreactive. And they can participate in the channeling of light, with the excitons that we discussed, taking to the reaction center. And in kind of like this, it's part of the antenna complexes. They also protect, as it turns out, the membranes of the thylakoid in the chloroplast from photooxidation. So it basically does what the way we like to think with healthy nutrition. And if you go to the health food section of any grocery store and you see beta carotene, you can buy it. They tell you antioxidant, but actually it is and does protect the membranes in the chloroplast. What about the 4 electrons? Okay, that we need here. Well, so how many light flashes do you need to release oxygen? I mean, this is a very simple. So we know that this is light catalyzed. All right, this reaction needs light to happen. Without light, it doesn't happen. So you just have to go giving them flashes and ask how many flashes does it take. And sure enough, it takes. So those are the so-called flash yield experiments. And you can see this beautiful, periodic pattern which repeats every 4 flashes. Okay, you have 4, 8, 12, 16, and then it loses synchrony at some point. So the reaction scheme is not entirely obvious. Okay, these are the suggestions. These are the 4 flashes that are coming in: 1, 2, 3, 4, and all the various stages of oxygen. And these are the protons that are coming off, and eventually oxygen comes off too. The only point about this scheme is that it is very heavily dependent on these metals. Manganese, okay, is very important, and manganese will it is thought to coordinate oxygen in this way, and that's why it's absolutely essential. And at this point, we will stop, and we will continue in the next video with photophosphorylation.